and today we're going to look at genetic variation, and we're also going to talk about meiosis. First of all, genetic variation is hugely important to evolution. Without genetic variation, populations won't be able to evolve. We might think of variation as the framework upon which evolution acts. Some possible sources of genetic variation, either in an individual or in a population, can be mutations. You may not have gotten there yet, but we'll talk about point mutations, which are when one nucleotide is incorrectly placed in a DNA sequence. We might also talk about frame shift mutations. And those are mutations in which extra nucleotides are either inserted or deleted from the DNA sequence. Random fertilization is another source of variation in populations. In sexual reproduction, a random sperm and random egg get together to form a new offspring, and that creates lots of genetic variation. Meiosis is another source. During meiosis, two important events occur that create variation. Crossing over happens in prophase 1. We'll talk about how that works in a minute. And also independent assortment, which occurs in metaphase 1. And a final source of variation that we'll mention is immigration. When new individuals join a population, they bring with them unique alleles and therefore provide the population with some variation. So now we're going to talk about meiosis. Meiosis is a specialized type of nuclear division of the nucleus, sort of like mitosis. It reduces the chromosome number in half. During meiosis, 4 haploid cells are created, and each one is genetically distinct from the parent cell that they came from, and also from each other. Let's go through some definitions to make sure you understand those before we go further. First of all, haploid cells. And notice N here. N really stands for number, and we could think of it as 1N because haploid cells are cells which contain only one copy of each type of chromosome. In the human body, the only haploid cells are the gametes, or the sex cells. Diploid cells, notice the symbol for diploid is 2N, and that means 2 of each kind of chromosome. These are your normal body cells, in humans at least. Most body cells are diploid. We call those normal body cells somatic cells, and we see that defined here. Normal body cells like skin cells, muscle cells, cells in your stomach, diploid cells in your body. There's an advantage for a cell to be diploid, and that advantage is that a lot of mutations are recessive. A lot of dangerous mutations are recessive. And if a cell is diploid, it's protected against those because more than likely one of its genes is normal if one is mutated. Gametes are sex cells, sperm or egg, and in humans those cells are haploid. They only contain one copy of each kind of chromosome, and therefore 2 haploid cells, when they go through the process of fertilization, create a diploid cell known as the zygote. And that cell will then undergo mitosis over and over again to form a new organism. One final term here are germ cells. Germ cells are the diploid cells, 2N cells, that are found in either the ovaries or the testes. They're the only cells in the body that undergo meiosis. These are the cells that eventually help to form the gametes, the sperm or the eggs. So some roles of meiosis in the body. First of all, it creates haploid gametes. It makes sexual reproduction possible. It also creates cells that are genetically different from each other. And like we said a minute ago, it can do that either through crossing over or through independent assortment, both of which occur very early in the process. So here's sort of an overview of the process of meiosis. If you understand the things on this slide, you understand most of what's here, what meiosis is about. So we start with a cell that's diploid. This is a germ cell. This cell is 2N equals 4. If this were a human cell, it would say 2N equals 46. There would be 46 chromosomes inside that cell. Between this first picture and the second picture, we could say interphase is happening. Specifically S phase of interphase, the DNA gets replicated, And notice that the chromosomes go from being one chromatid to two chromatids. They go from they become bivalent chromosomes. Chromosomes consisting of two chromatids. Notice this still says 2n equals 4. In a human, this would still be 2n equals 46. If this were a human cell, you would see 46 X-shaped chromosomes there. The meiosis really occurs in two major stages, two major parts, meiosis I and meiosis II. Meiosis I is often called reductive division because meiosis I takes a diploid cell and from that diploid cell creates two haploid cells. So notice what happens in meiosis I. The homologous chromosomes, and that's another important word, homologous chromosomes. Sometimes you'll see them called homologs. Those are the chromosomes of the same type. So they have the same genes in the same order on those pairs of chromosomes. They may have different versions of those genes. They may have different alleles, but they control the same traits. In meiosis I, the homologous chromosomes line up, and then one set gets pulled into one of the daughter cells, and the other set gets pulled into the other daughter cell. So after meiosis I is over, we have two cells, each of which is now haploid. And here we say n equals 2 in these pictures, but in a human, this one would be n equals 23, and so would this one. Then you get meiosis II. These cells that resulted after meiosis I really aren't ready to work yet because the chromosomes are bivalent. After any kind of cell division, whether we're talking mitosis or meiosis, if it's over, the chromosome should be made of a single chromatid. These chromosomes are not. They're still bivalent. So meiosis II's job is to take the bivalent chromosomes and to segregate the chromatids from each other. So both of these cells undergo meiosis II. Meiosis II essentially works just like mitosis. There's no DNA replication between meiosis I and meiosis II, so the cell split. We end up with 4 cells. All of them are haploid. In a human, these cells would all be n equals 23. And all the chromosomes are made of one chromatid, not two. These cells will undergo a period of maturation and will eventually become the gametes. All right, this slide essentially walks us through the life cycle of people. Each human is diploid. Again, remember that means that there are 2 copies of each chromosome in each cell. In the reproductive organs, meiosis occurs, and diploid germ cells are used to create haploid gametes. Reproduction occurs. Haploid sperm and haploid egg get together during the process of fertilization to create a diploid zygote. So each of these gametes would have 23 chromosomes, and 23 plus the 23 would give us a zygote with 46. That zygote will then undergo mitosis thousands and thousands of times and grow into an offspring, and then the process will repeat. So let's talk about the stages of meiosis. First of all, the germ cell is going to spend a big part of its time in interphase. Interphase is not a stage of meiosis. It's not a stage of mitosis. It's what occurs when the cell isn't dividing, when it's going about its normal daily routine doing its job. Remember that it consists of G1, where the cell grows, S, where the DNA is synthesized or copied, and G2, where the cell continues to work and prepare for cell division, organelles are copied, and so on and so forth. But remember, S phase is when the replication of the DNA occurs. After S phase has happened, the germ cell will now, it's always a diploid cell, but it's a diploid cell now, and if you could see them, all the chromosomes would be bivalent after S phase of interphase. As I said a minute ago, meiosis I is known as reductive division because it's going to start with one diploid germ cell, and by the time it's over, you'll have 2 haploid cells. And those 2 haploid cells will have bivalent chromosomes. at the end of meiosis 1. Chromosomes that consist of 2 chromatids, like so. So in meiosis 1, the very first actual stage of meiosis is prophase number 1. You do need that Roman numeral. It's very much like prophase and mitosis. During it, the nuclear envelope disintegrates. The chromatin, the DNA, will condense. It'll wind up, and it'll form the chromosomes. They'll become visible. The centrosomes will start to make the little spindle fibers that will be used to pull around the DNA. But in addition to those normal prophase events, a couple of extra things happen in meiosis 1, or prophase 1 of meiosis 1. First of all, you get synapsis. This is when homologous chromosomes of the same kind, attach together, and they form this setup called a tetrad. It's called a tetrad because it consists of 4 chromatids. While these chromosomes are hooked together, we get crossing over. Crossing over is essentially when homologous chromosomes exchange might be a better word, segments of DNA. So that when it's over, the chromosomes that you end up with are different from the ones you started with. It's almost like shuffling a deck of cards. It's a way to reshuffle genes. We might think of it as a type of genetic recombination, and it allows a parent to pass chromosomes to the child that parent doesn't actually have themselves. So it's a hugely important way of creating genetic variation. So here we see a cell that's probably in interphase. Remember, actually in interphase, we couldn't see the DNA because it's going to be in the form of chromatin. This cell is a germ cell. It's 2n equals 4. Remember, in a human, it would actually be 2n equals 46. You should be able to see 46 single-stranded chromosomes. Again, if you could see them. DNA replication happens, so that's the S phase of interphase. And those chromosomes go from being single-stranded to being made of 2 chromatids each. And then we get meiosis 1 and prophase 1. Remember, in prophase 1, the nuclear envelope disintegrates, the spindle fibers are formed by the centrosomes, the DNA starts to become visible, and the homologous chromosomes synapse. They join together. In this picture, different colors are used to show you that one of each pair comes from each of your 2 parents. So here we see the points at which these homologous chromosomes are attached. You'll hear these points where they're attached called chiasmata. And when they're unattaching at the end of prophase 1, sometimes the chromosomes break and pieces are switched between the 2 homologous chromosomes. So what results are chromosomes that look like this. Chromosomes that are combinations of the actual chromosome that the parent has. These possibly could be passed on to the children. So after prophase 1, we get metaphase 1. And if you're calling mitosis, in metaphase, essentially the chromosomes are pulled to the middle and they line up single file along what's called the metaphase plate, the middle of the cell. Something similar happens here. Here, the homologous chromosomes line up across from their homologous partner along the metaphase plate. Something important happens here called independent assortment. We'll come back to that in a second. But here's a picture sort of what metaphase 1 might look like. Homologous chromosomes are lined up. Now notice in this particular picture, all the chromosomes from one parent are lined up on one side, the red ones, and all the ones from the other parent are lined up on the other side, the blue ones. That could happen, but in reality, they could line up this way, or this way, or this way, or lots of different ways. The side that a particular chromosome lines up on is random. This particular cell has 3 pairs of chromosomes, and that means that they could line up in pairs like this, 2 to the 3rd different ways. So there's pairs of 2, they could line up. There's 3 pairs of 2, they could line up 2 to the 3rd ways, that's 8. So there can be 8 possibilities. For a human, We have pairs of 2, but we have 23 of them. And if you figure this up on your calculator, you would say it's somewhere between 7 and 8 million different ways. So each one of us, exclusively because of independent assortment, can create 7 to 8 genetically different gametes. So let's go back and give a more formal definition for independent assortment. It's when the maternal and paternal chromosomes line up on opposite sides of the metaphase plate across from their partners in a random fashion. Again, it's sort of like shuffling the genetic card deck. What's going to happen after metaphase is anaphase 1. And in anaphase 1, essentially the little spindle fibers will shorten, and a member of each homologous pair is going to get pulled toward each side or each pole of the cell. So they've lined up, and now they've been separated from each other. Then we get telophase 1. Telophase 1 is a lot like telophase in mitosis. Essentially, it's the reverse of prophase. So the DNA makes it to opposite sides of the cell. A new nuclear envelope forms around each set of DNA. Spindle fibers are taken down. The chromatin will unwind, or the chromosomes rather will unwind back into chromatin, and cytokinesis happens. Remember, cytokinesis is when the actual cell or the cytoplasm is split in two. So meiosis 1 ends with 2 cells from the original one. The original cell was diploid. These 2 cells are going to be haploid. But the problem, what's not quite right about these 2 cells, is that the chromosomes in there are still bivalent. They're still shaped like X's. And we don't need that. We need our chromosomes at the end of cell division to be just a single strand, a single chromatid. So because of that, we're going to end up with meiosis II. Before we get to meiosis II, here we see metaphase 1. Homologous chromosomes are lined up across from their partner. Notice that these 2 down here have underwent crossing over in prophase 1. You can see that this has a little blue piece and this has a red piece. That's trying to tell you that the red one has genes from its homologous blue partner, and the blue one has genes from its homologous red partner. Anaphase 1, the spindle fibers shorten and pull this set of DNA this way, and this set of DNA this way. Next, we get telophase 1. And again, in telophase 1, it's essentially the opposite of prophase. The DNA makes it to opposite sides. There's a new nuclear envelope formed around each set. The chromosomes will unwind and go back to being chromatin, and we'll get cytokinesis, which will split this cell right down the middle. So end up with 2 cells. 2 cells that are haploid, but haploid with bivalent chromosomes. We get a period of interkinesis, sort of like interphase, but what does not happen during interkinesis is that there is no DNA replication, because we're trying to lower the amount of DNA. It would be counterproductive to copy it here. So then we get meiosis II. And meiosis II is pretty much just like mitosis. During meiosis II, both of the cells that we got from meiosis 1 are split again. And this forms 4 cells. 4 cells that are haploid, and they all come from that original germ cell. These haploid cells are going to have chromosomes formed of only 1 chromatid. These are the cells that will, after a period of growth and development, will become the gametes, the sperm or the eggs. The stages of meiosis II are prophase 2, metaphase 2, anaphase 2, and telophase 2. A lot of people use the acronym PMAT to remember the order of the stages. So in meiosis II, here we see both of the cells that resulted from meiosis 1. Remember, they're both haploid. So these cells enter prophase 2. The chromatin condenses and becomes chromosomes. The centrosomes form the spindles. The nuclear envelope is going to disintegrate. The nucleolus will go away. Then we'll get metaphase 2, where the chromosomes line up along the metaphase plate in the middle. Here they line up single file. because they don't have any partners anymore. There are no homologous chromosomes in either of the cells. Those were done away with in part 1. Then we get anaphase 2. In anaphase 2, the spindle fibers again shorten, but this time they pull apart the bivalent chromosomes into single chromatids. This set of DNA is pulled this way, and this whole set is pulled this way. And that happens in both cells. Eventually, the DNA makes it to opposite sides. We get a new nuclear envelope around each set of DNA. Spindle fibers are dismantled. The chromosomes unwind, go back to being chromatin. Cytokinesis happens. This cell splits. So does this one. We end up with 4 cells. 4 cells that are haploid, that have chromosomes that are made of a single chromatid, and they're all genetically different from each other because of things like crossing over and independent assortment. And again, these cells will grow up to become the sex cells, the gametes, or the sperm and eggs. And in humans, they're all going to be N equals 23. So let's also talk about gametogenesis. Meiosis is a part of gametogenesis. Gametogenesis is essentially meiosis, followed by a period of growth and development. Gametogenesis literally means the creation, genesis, of gametes. So in males, you're going to hear it called spermatogenesis, the creation of sperm. And in males, one diploid germ cell goes through meiosis, pretty much just like we described it, to make 4 haploid cells, and those all grow up to become sperm. In females, you're going to hear it called oogenesis. And in it, one diploid cell splits unequally to produce 4 haploid cells. Only one of those ends up being large. That large cell ends up becoming an egg. The small cells are called polar bodies, and they usually just die. And we'll talk about why it occurs this way in a minute. So here we see the process for men. This is spermatogenesis. So this is our germ cell. This cell is diploid, and the chromosomes all are going to be bivalent after at least S phase. So we get meiosis 1. And remember, meiosis 1's job is to split the homologous chromosomes from their partners. So what we end up with, let's say this is in humans, so this would have 46 bivalent chromosomes in it. After meiosis 1, each of these cells will have 23, but the chromosomes are still bivalent. Remember that meiosis 1 is called reductive division because it reduces the chromosome number in half. Meiosis 2 happens, and remember its job is to separate the 2 chromatids that make up each chromosome, each bivalent chromosome. So that happens, we end up with 4 cells. Each of these cells is still going to have 23 chromosomes, but instead of being bivalent, they're going to be made of just one piece, one chromatid. Then we get the period of growth and maturation, and we end up with our mature, in this case, sperm. 4 sperm from 1 germ cell. And here's the way the process works in females. Oogenesis. So this cell, the yellow one in the middle there, that's the germ cell. That cell has 46 chromosomes, and after S phase of interphase, they're bivalent. Notice there's some other cells that sort of surround it, but here we're looking at this purple nucleus. Meiosis 1 happens. And most of this happened before birth in females. The germ cell splits, the nucleus does, and forms 2 cells. One large, one small. These both now have 23 chromosomes, and they are bivalent still. The small cell is called the first polar body. It's haploid. This other cell will become the egg eventually. It's called the secondary oocyte. If a sperm happens to penetrate one of these cells at this point, it will undergo meiosis 2. Otherwise, if a sperm doesn't penetrate it, it just dies. So let's pretend that a sperm does penetrate it. The other cells, the big cell will split again. This first polar body may also split. We may end up with 3 polar bodies and 1 large cell. All of these nuclei are going to be N, so they're going to be haploid. In humans, they would have 23 chromosomes in them, and those chromosomes now would be made of just 1 chromatid. The polar bodies die. That 1 cell ends up becoming an egg. The sperm nucleus fuses with it, and we end up with a zygote. It got an N nucleus from the sperm. It got an N nucleus from the egg. So that nucleus is now a diploid nucleus inside the zygote. Now let's think about why in females it happens this way. And the main reason is what the female provides to the baby. The male parent provides 23 chromosomes, half of the genetic material. In addition to doing that, the female also has to provide food. It takes time for things like the placenta, the umbilical cord to form. In the meantime, the developing zygote has to feed off of food that's stored inside the egg. So to keep that zygote alive, the egg has to be big and it has to be filled with nutrients. That's the main reason you only get 1 working egg from each germ cell. So now I've got some questions to look at. First of all, a diploid cell with 50 chromosomes undergoes mitosis, not meiosis, and cytokinesis. Mitosis and cytokinesis produce which of the following? So our cell is diploid. It starts with 50 chromosomes. Remember that mitosis takes 1 cell and ends up creating 2. So we know our answer's got to be either A or B. Remember also that mitosis is cloning. So the cells that you start with should be exactly like the 2 cells that you end up with. So we're going to get 50 in each of the daughter cells. And remember that whether we're talking meiosis or mitosis, if it happens correctly, each chromosome should be made of only 1 chromatid. So the correct answer here that we're looking for is B. Here's question 2. During meiosis, one blank cell produces blank haploid blank. Well, here's the thing. We're starting with a diploid cell. And that diploid cell is called the germ cell. And it's going to produce 4 cells that are haploid and that grow up to become gametes. So what we're looking at here is answer C. Question 3. At the end of meiosis 1, what is produced from 1 diploid germ cell? Remember, we start with a diploid germ cell. It undergoes meiosis 1. Meiosis 1's job is to separate the homologous pairs from each other. So if this thing had 46 chromosomes, 2 sets of 23, each one of these 2 cells would end up with 23 chromosomes each. Remember, though, that if these cells were bivalent to begin with, they're still bivalent after meiosis 1. So the point being here is we end up with 2 cells that have half as many chromosomes as the original cell. Chromosomes are still bivalent, so we're looking at answer B. Next one, during which stage of meiosis does independent assortment occur? Remember, independent assortment is when homologous chromosomes line up across from their partner along the metaphase plate, and they do so in a random fashion. That happens in metaphase 1 of meiosis. Here's question 5. Crossing over occurs in the very first stage of meiosis. That's prophase 1. And remember that crossing over is when homologous chromosomes actually exchange genetic sequences and they essentially recombine the genetic material, an important source of genetic variation. Question 6. How many functional oocytes or eggs are created from 1 germ cell in a human female? Remember that in females, one germ cell ends up making one working egg and 2 or 3 polar bodies. The answer here, though, is you only get one functional egg cell. Here's question 7. It looks very much like question 1. This time we have a cell, diploid cell, with 50 chromosomes. It undergoes meiosis and cytokinesis. And we want to know what we get. Well, we know that in meiosis, one cell ends up creating 4. This cell is diploid. These cells should be haploid. This cell should have chromosomes that are bivalent. These cells should end up with chromosomes that are made of one chromatid. So 4 cells with half the number of chromosomes of the original cell, so it should be 25, and each one should consist of only one chromatid. So we're looking here at answer D as our correct answer. All right, now we're going to talk a bit about using crossover frequencies to determine the order of linked genes. All right, first of all, some important terms. Linked genes refer to genes that are found on the same chromosome. So let's say we had some genes that were very close to each other. For these two to cross over within the homologous chromosome, the chromosome has to break somewhere between them so that they can swap over. Now let's look at this gene and this gene right here. They're much further apart. And that means there's more places where this chromosome could break and they could potentially cross over. The point being, the further two genes are apart from each other, the more likely they are to cross over to the other homologous chromosome during prophase 1. So we can use the frequency of crossover almost like an estimate of length. The more two genes cross over, the further apart they are, is essentially where we're going. We can use that to do what's called gene mapping, to essentially determine the relative order of the genes on a chromosome, the relative order, let's say, of linked genes on a chromosome. So let's see what we got here. We've got a listing of genes A, B, C, D. They're linked, they're on the same chromosome, and we're looking at how often they cross over, their crossover frequency. What I would recommend for you to do in a problem like this is to take the two that seem to cross over the most, write those down, like so, and go ahead and label that they cross over 75% of the time. And that means if they started off on the same chromosome, these two genes 75% of the time end up on the other chromosome. They cross over that much. So now you want to find one of these genes that's close to either gene A or gene B, and you know it's close because it doesn't cross over with it very much. That would be gene C. Gene C could be located here, but it just as easily could be located here. This is a crossover 15%. So it would be nice if we had a comparison between C and B. But notice they didn't give that to us because it would make it too easy. So now we need to look at gene D. And if you look at gene D crosses over with A 40% of the time. It crosses over with B 35% of the time. Well, what I'm hoping that's telling you is that D is somewhere between A and B, and it's a little closer to B than it is to A. So gene D is somewhere in here. It crosses over here 35% of the time, and it crosses over here 40% of the time. Well, now we can compare C to D to determine if C is actually here or if it's here. Notice that C and D cross over 55% of the time. Well, that tells you that C can't be here in the middle, because the crossover between A and D is only 40, but the crossover between A and C is 15. Well, I'm hoping you see that that means that these two should cross over 55% of the time. So what we've just done is we've mapped this chromosome. We know the relative order of the genes on it. A possible answer could be C, A, D, B. Another possible answer could be B, D, A, C. We just flip-flop the order. Because this doesn't tell you the order, this doesn't tell you where they're at on the chromosome, it just tells you what's next to what. It's not an actual like measurement of length. It's an estimation of it, but it tells you the relative order of the genes on the chromosome. And in this case, our answer is either CADB, which notice is not one of our choices. The correct answer here is BDAC. Let's look at one more example of crossover frequency problems. So here we have determine the sequence of genes along a chromosome given the following recombination frequencies. A and B crossover or have 8 map units. Sometimes instead of telling you the percentage of the time they cross over, they give you this unit called a map unit, which, like I said, they're using almost like an inch or something, like a unit of measurement. Each map unit, though, is equal to 1% crossover. So this is no different than the last problem we worked. So as I said before, we want to find the two that appear to be the most far apart, that cross over the most often, and that would be B and D. Write them out like so. They cross over 33 map units, or 33% of the time. Try to find a gene that's near either side. Well, A and B only cross over 8 map units. So A could be here, or A could be here. Now, here they were nice to us. They gave us a comparison between A and D. Notice A and D only cross over 25% of the time. Well, from B to D is only 33. That means A cannot be here. A's got to be here between the two. This is 25 map units. And A to B is 8 map units. And notice that 8 and 25 add together to be 33. In a real world problem, they may not add to be exactly 33. They might add it to be 33.6 or 34, but they should add up to be close. We're still missing a gene. We're missing C. So A and C cross over 28% of the time, and B and C cross over 20 map units. So we need to figure out where it's at, and it looks like C is going to be, C has to be over here because A and C cross over 28% of the time. B and C only cross over 20% of the time. So our answer on this particular problem is CBAD, or same answer, DABC. Let's go ahead and look at one more. So this time, again, we have 4 genes. This time they're J, K, L, and M. They're linked. They're on the same chromosome. We have the the crossover frequencies. So we want to find the two that seem to be the most far apart, which are going to be J and M. They cross over 12% of the time, 12 map units. Find one that seems to be close to either end, and that's going to be K. K could be here, or K could be here. We need to figure out which side K is on. So it would be nice if we had a comparison between K and M. We do not. So let's look at J. Actually, let's look at L. L and M cross over 7% of the time. L and K cross over 8%. So that's telling me that L has to be somewhere between J and K. So we're going to put L somewhere right here. L and M cross over 7% of the time. Well, that's telling me if J to M is 12, then and L to M is 7, then L to J has to be 5. Well, if L to J is 5 and K to L is 8, K can't be here. K has to be out here. So K and L cross over 8% of the time. J to K is 3. So we've now mapped this chromosome. Our answer is K, J, L, M, or M, L, J, K. All right, I hope that helps.